अब हम बात करेंगे स्टेप बाय स्टेप प्रोसीजर ऑफ डीएनए आइसोलेशन की। सबसे पहले वी हैव टू बी श्योर all the required reagents, buffers, stock solutions are available. अगर कोई चीज़ अवेलेबल नहीं है या required volume में मौजूद नहीं है, we have to prepare them so that during the procedure we should not be looking for some missing components. Whenever we have to isolate the DNA from any cell, we have to destroy the outermost layer of that cell. As we are talking about the bacterial cell, in this case we have to remove its outer cell wall and then cell membrane so that the cellular components are made available. But at the same time हमें इस चीज़ का ख्याल रखना है कि सेल वॉल ब्रेक करते हुए हम प्लाज्मेट को ब्रेक मत कर दें. For this purpose, if our intention is to isolate the plasmid, we have to carefully treat the bacterial cell with the lysozyme in the presence of EDTA and sucrose solution so that its cell wall gets partially damaged, and then we are able to treat it with the Triton X100, which is a detergent that can later on destroy the cell membrane, keeping the plasmids intact. As you can see in this picture, this is the partially damaged cell wall. This structure is called spheroplast, and once this spheroplast is produced, Triton X100 can damage the plasma membrane, leaving behind the cell extract containing the pieces of cell membrane and rest of the cellular components, which later can be centrifuged. All the cellular debris, large DNA fragment from the genomic DNA, they can be settled at the bottom, leaving behind the solution which only carries the intact plasmid. The procedure is going to be step by step. The first step would be the culture transfer, and here we have to maintain the sterilization using aseptic method, 1.5 milliliter. As we are working with the mini prep, so small volumes are required, will be transferred to sterilized microcentrifuge tube. We have to harvest the bacterial cell. For that purpose, centrifugation is done at 14,000 rpm in the microcentrifuge for 1 minute at room temperature. By this step, all the bacterial cells, they are going to settle down at the bottom, and we can aspirate the culture media that is no more required at this moment. As we have to proceed further, we have to make it sure that the kind of washing is done, all the unwanted components are removed. We will give an additional 1.5 milliliter of culture media to pellet. We will resuspend it in the pellet. The bacterial pellet is going to be resuspended in the culture media, and again the centrifugation step at number 2 will be repeated. So centrifugation at 14,000 for 1 minute and aspiration of the culture media, leaving behind the pellet. We have to dry this pellet, and now we start using the buffers to isolate the plasmid DNA. This pellet will be resuspended in lysis buffer 350 microliter or 0.35 milliliter, and by vigorous verticing we are going to break the pellet, or we are going to mix the bacterial cell with the lysis buffer. As the name indicates, this buffer will help in the lysis of the bacterial cells. At the same time, we have to add some lysozyme and very partial vertexing, because this is an enzyme. It will be quick in performing its function. So immediately after the given time period, we have to inactivate the lysozyme, and this inactivation is achieved by incubating the tubes in styrofoam in the boiling water bath for 40 seconds. So I repeat here that inactivation of lysozyme is going to be achieved by incubating these tubes at boiling water bath for 40 seconds. Now the next step is again centrifugation for 10 minutes at 14,000 rpm at room temperature. This centrifugation is purposed to remove all the cell debris, the lysed cellular components, leaving behind the plasmid DNA, because the lysozyme treatment has already damaged the cell wall of the bacteria and these cells were ruptured. As the product of the centrifuge is the pellet, this pellet is of no interest, so we are going to remove the supernatant, or the other way, using toothpick we are going to remove the pellet from the bottom of the plastic tube. The next step would be precipitation of the DNA. Our supernatant is now containing our plasmid DNA that needs to be precipitated. For that purpose, we are going to use sodium acetate and isopropanol, which will help in precipitation of the DNA, vertex mixing, and then holding it 20 minutes at room temperature will facilitate the precipitation. Again, centrifugation at 4 degrees centigrade, 14,000 rpm for 15 minutes, and removing the supernatant, as the DNA has already been precipitated, is already settled as a pellet on the bottom. Then removing the supernatant and letting the pellet dry. And now this is our DNA, which will be resuspended in TE buffer that should be free of any kind of DNase enzyme. And after resuspension, this DNA will be stored at 4 degrees centigrade for further usage.